So hello everybody. I would like to get your attention please and start the second session of this morning. I'll put a bit more force. Yeah, that's the next phase.
Sorry, Matt.
Yeah, no problem. Yeah.
I was too intensive in the discussion.
Yeah. So it's a good thing to see that the discussion already is starting to take form. So please save all the topics that you would like to discuss for the plenary session. So for now, we have 2 more talks before we actually have the first discussion session. And after this discussion session, we'll break for lunch. So this morning's session, we've been hearing from Sebastian and from Adrian on application in virology. Gilles talked to us about applications in bacteriology. And now we have Jamie Cubero from INIA on the stage, who will also be talking about an application of genomics tools for xanthomonads.
No. Good morning, everybody. First, thank the organizer for inviting me to give this presentation and show you some of the results we have on Xanthomonas, mainly on Xanthomonas arboricola. But I have to say one thing, that we are, in our group, I like to say that we are plant pathologists. We are not molecular biologists, we are not geneticians, we are plant pathologists. And as plant pathologists, we have to provide tools for disease control. And this is what you will see during my talk. I'm not going to show you so many results. Just I will try to convince you about the utility of genomics for plant pathology and for disease control. Well, this is a brief overview of what you will see during my talk. I'm going to talk a little bit about the past problem on Xanthomonas. If I have time, I will try to show you some ideas or what we should do, or my opinion, what we should do in some other model in different Xanthomonas. And in most of the talk I'm going to talk about Xanthomonas arboricola patubarprunite, that is a quarantine disease in the European Union and is an important plant pathogen now in Spain. Well, during my talk, you will see, I told you that we are plant pathologists. You will see plants, and you are going to see also symptoms, because I like to say to my student in the lab or to my group that we are not doing genomics for nothing. You are not doing genomics for academic purposes. We are doing genomics for something useful and for something that can be used immediately. Well, we are working in a group of plant pathogenic bacteria, the genus Xanthomonas. This is a really interesting group for microbiologists and also for plant pathologists. This is a group of bacteria that show a high diversity, and not just only as a high diversity in terms of genomics or in genetics, but also a high diversity because they are able to infect different varieties of host plants, and they show different methods for infection, colonization process, and they show different diseases in different hosts. These are some of the most important crops affected by Xanthomonas. We are going to talk about Xanthomonas citrus subspecies C3 that is affecting citrus crops, and also about Xanthomonas arboricola patubarprunite, that is the bacterial spot of stone fruit. It's affecting peaches, almond, apricots. But there are also different Xanthomonas affecting, for instance, rice, tomato and pepper, and also brassica plants. When we talk about Xanthomonas, most of the time we talk about pathogenic bacteria, but sometimes we don't realize, in the past we didn't realize that in addition to these pathogenic bacteria, we are able to find in plants some Xanthomonas that are not producing any symptoms. And you may wonder, why this guy is talking about something that is not doing anything in the plant and is not causing any disease. Well, this is important in terms of diagnostic because sometimes these bacteria were misidentified and were considered pathogenic, and this meant something. You will see in next example. I'm going to talk to you about one example on citrus Xanthomonas, and something that happened several years ago in Florida. Well, in citrus, there are two different types of Xanthomonas that are causing disease. One is citrus bacterial canker, that was considered one of the most important diseases in citrus. It's an important disease. Just right now, maybe it's not so important because there are some other things like Hualon bean or Xylella that are affecting citrus are considered much more important in the areas where they have the disease. And these are lucky guys in Florida, for instance, or in Brazil, have citrus canker and HLV, and of course they consider HLV much more important and more serious than citrus canker. But at the beginning, this disease was considered a really, really serious disease in Florida. Citrus bacterial canker is caused by Xanthomonas citri subspecies citrae and is causing this kind of lesions. But there is another disease that caused by Xanthomonas that is citrus bacterial spot that is caused by Xanthomonas alpha-5 subspecies Citrumilonis. Don't take too much attention to the name because in Xanthomonas we change the name almost every day. This is a cause of citrobacterial spot that is not a serious disease. It's just causing some symptoms in leaves. That is not. Even in young trees, it's affecting young trees, but it's not affecting adult trees, and it's not affecting the fruit. That this is the most important in citrus canker, that is affecting the fruit. Well, this is a long history in US, in Florida. In Florida, this is citrobacterial spot. Well, you see, this is a different name, but it's the citrobacterial spot. In 1984, they identified this disease mainly in greenhouses, in nurseries, and because these kind of lesions, they consider this disease as citrus bacterial canker. Because was a citrus bacterial canker, was considered citrus bacterial canker, was subjected to the rules of citrus canker, and this means that they burn plants with the bacteria. They destroy the trees with the canker symptoms within the orchards and the foliage from the trees. And the fruit from these trees could not be used for fresh market. The fruit could be used just for use, for processing. Well, this is everything, all of this is here in this nice paper from people from Florida. These rules that, because they consider citrus canker, about 20 million trees were destroyed, and this cost about $90 million. This is a huge amount of money, and this is a high economic losses. But finally, in 1990, they decided that this was not citrus canker. You remember that this started in 1984 and finished in 1990. This means that they spent a lot of money in something that probably they didn't need to use. All this was necessary. Well, the answer is now clear. It was not necessary for sure. It was not need to eradicate all these trees because it was not citrus canker, and the bacteria are not doing almost anything in the tree. Was a mistake? It was not a mistake. I mean, they take this decision based on the knowledge of the bacteria they have at this time, and they have to do that. With genomics, this could be solved. Well, this is a slide I used to put when I was working in Florida. When in Florida they have panthers and they have cats also, and you can find a panther in your garden, and you can find a cat in your garden. If you find a panther, you are going to be in trouble, because not because the panther is going to do anything else. It's not going to do anything. It's because it's a protected species, and you're going to have problems. And the services in Florida are going to go to your garden and going to retire the panther. If you have the cat, probably even you are happy, and you're going to adopt the cat, and you're going to live forever with the cat. It's the same with the citrus canker. If you had a citrus canker at this time, the plant protection service was going to go to your garden and gonna remove the tree and all the trees around you, your infected tree. If you have the citrus bacteria spot, doesn't care. I mean, nobody care about that. Well, this is something clear. If you know the enemy, well, this is something that is really smart from this guy in China. If you know the enemy and you know yourself, you need not fear the result of 100 battle, and that's the same in plant pathology. The first thing you need to do is to know which is your enemy and what is doing your enemy. And this is knowledge can be acquired by genomics. With genomics, you can have a precise identification of the causal, of the disease causal agent, and also you can understand, or you can try to understand which are the mechanisms that are involved in that make this micro pathogen, and why this pathogen is doing what they are doing on plants. And this is another part. I'm not going to talk about this, but another part is with this new next generation sequencing. You can not just know if your pathogen is in the plant or not in the plant, but also if the pathogen is surrounded by other microbes that can help to the pathogen or can go against the pathogen. I mean, the microbiome is involved also in plant health. Well, this is, I told you that it's a past history that occurred in US several years ago, but you will see that we have also similar thing, not so bad result, with not so bad result, with Xanthomonas arboricola patobarpurnai. But thanks to God, at this time, we have genomics, and we could solve the problem from the very beginning. I told you that I'm going to show you some symptoms, some plants. Well, this is a bacterial spot of stone fruits. It's producing this kind of symptoms in leaves and also in fruits. It's still a quarantine bacteria in the European Union. And as citrus canker, the presence of this disease involves many economic losses in a country. Because this fruit, of course, you are not going to buy this fruit in the market because you don't like this kind of fruits. And for us, it's especially interesting because it's affecting also almond. Well, the disease is spread almost everywhere in the world. They have stone fruits. And in Spain, the situation is like this. We have the disease in many hosts, in plum, in apricot, in almond. Yeah, and for us, it's really interesting that because we have the disease in almond. When we start to work with this model, we were encouraged, at this time we were working on citrus bacterial canker, but we were encouraged to work on this disease because it was identified in Spain. And because we were working in Xanthomonas, they told us, okay, do something with Xanthomonas arboricola patobarpurnai. And the first thing we did was to make a collection of strains that were identified, were isolated from the plant protection services. Of course, the first thing we did was do a very simple analysis, with multi-locus sequencing analysis, and we suddenly realized that most of the strains that were provided by the plant protection services were clustered with those strains that were our reference for Xanthomonas arboricola patobarpurnai. But a few of them, a few strains, were misidentified. And this, we found some of the strains that the plant protection services considered were Xanthomonas arboricola patobarpurnai, and at least in our first assays, we couldn't consider like this. And these guys were doing a PCR based on to identify the strain. We're using a PCR reaction based on an ABC transporter that is a transporter that is present in many bacteria, and they consider this ABC transporter as specific. You will see, I will talk later a little bit about this ABC transporter. But the first thing is to say is that this strain were misidentified. I mean, were not really Xanthomonas arboricola patobarbonae, even the positive result with the first PCR reaction. Of course, the first thing we did was to characterize phenotypically this strain. We inoculated in several hosts, and this strain didn't produce any symptom in any of the hosts we test in all the prunus species. And even we inoculated in different hosts to try to see if they were able to produce any kind of symptom in other hosts different to prunus species. We also characterize different. We are really interested in some mechanisms of the bacteria to produce the disease, especially in those first stage of the infection process, and we test motility, chemotaxis, and we also did several biochemical tests, and these strains were different to the Xanthomonas arboricola patobarbonae. Of course, we thought, okay, we're going to sequence all this strain, and this is the result, the first result we have with a few of these strains, and we realized that a big difference between pathogenic and non-pathogenic strain. The pathogenic strain has this plasmid, and the other strain has no plasmid. This plasmid is described to confer virulence to the strain, but well, this clearly indicated that the strain were different than those considered pathogenic. We also analyzed the core genome, and again, we could separate. Here we have the non-pathogenic strain, and here we have the pathogenic strain that cluster very well with not just with patobarbonae, but also with the more virulent pathobars in the same species, in the species Xanthomonas arboricola. I mean, it was common sense. I mean, this based on the core genome, we can separate the non-pathogenic from the pathogenic strain. Well, if you go in deep in the genome, again, this is similar to the result that Gilles presented today on Salrastonia. You find that just a few genes are specific for the pathogenic strain compared with the non-pathogenic strain. I have to remind you that these are Xanthomonas, and all Xanthomonas are taxonomically close. I mean, you are not expecting to have too many differences. With these 451 genes, we analyze and we try to find why one strain was pathogenic and why the other strain was not pathogenic. And as I told you, we are studying infection mechanism, initial step of the infection and advanced step of the infection, and we have found a difference in both sides. The bacteria have different behavior depending of if it's pathogenic or it's not pathogenic. I'm not going to go in detail on that, on all of this, but we have a difference on sensors, environmental sensor. I mean, the pathogenic bacteria sense different than the non-pathogenic. We have also a difference in mechanisms for bacterial attachment to surface and to recognize the plant cells. We have no much difference on flagella, for instance, for motility, but this was interesting because we have a small difference in one of the proteins of the flagella that is involved in the immune response to the plant, in the response against the bacteria. We have also difference in what the bacteria secreted to the plants. For instance, in the type 2 secretion system, we have difference in enzymes that degrade the cell wall of the plant. But the most important is that we have also difference in the type 3 secretion system. The type 3 secretion system is a secretion system that is involved in the virulence of not just plant pathogenic bacteria, but also clinic bacteria, human bacteria, animal bacteria. And this is a clear element for the virulence of the bacteria. And this is the tree, if you remember, this is the tree we had with the core genome. And we found that the pathogenic strain have the type 3 secretion system with 21 effectors of that was secreted by are secreted by the bacteria. We have some of the non-pathogenic strains that have no the type 3 secretion system and no effector. Well, lately some group here in France found one new effector for this strain, but it's not clear if this is an effector or is functional or not. But the interesting thing is that we have some strains that even with the type 3 segregation system have a few effectors, and these effectors are not enough for the bacteria to be pathogenic. Well, this is a good model because based on this different content, type 3 segregation system, effector, and system, you can figure out how evolve the bacteria, how acquire the virulence, and it's possible that these bacteria at the end are going to acquire some of the effectors from here and be again be pathogenic at the end. But the important thing here is that we have effectors that confer virulence to these bacteria, and these effectors, well, this is a summary of what we found. I told you I'm not going to go in detail, but these are, if you realize, there's a few differences between pathogenic and non-pathogenic strain. Well, I'm coming back to the method that these plant protection services were using for detection and for diagnosis of the bacterial spot of stone fruit. At this time, they were using an ABC transporter. If you look for this ABC transporter in other Xanthomonas, you're going to find this ABC transporter in other Xanthomonas. And of course, you can find this ABC transporter in non-pathogenic strains. And this was the problem. You have the ABC transporter even if they are not pathogenic, and that's why they misidentify this strain and they consider this strain as pathogens and were not pathogens. Well, even more, ABC transporter are a large superfamily of proteins found in all living organisms, including bacteria, archaea, and eukarya. This means that it's not a good target to try to detect a pathogenic bacteria. And most important, this is a quarantine bacteria. This is it's a mistake. It's like Cystobacteria sporis. It was a mistake? No, it was not a mistake. At this time, we had no information about the genomics of this bacteria, and was a good approach at this time. I mean, even in most of the cases, it was okay. I mean, they were detecting pathogenic strain, and they were considered okay, the pathogenicity. But, well, when we were doing the genomics, appeared this paper that this nice, very nice paper that these people identify effectors in Xanthomonas arboricula in different species. And one of the effectors is, I don't know if you are going to see, but one of the specific effectors was for Xanthomonas arboricola patobarpunae with the SOP3, that is a T3 effector. This is one of the effectors we already identify also by genomics. And this T3 effector, of course, you need to check if this effector is present in other Xanthomonas, and sadly, we found this effector also in other Xanthomonas species, but as different, the difference between this effector and the ABC transporter and this effector is always in pathogenic strain. In pathogenic strain that are affecting different crops, not just prunus species, but in different crops, but this effector is for instance present in Xanthomonas oryzae that is affecting rice. But is always present, never present in Xanthomonas which were not described as pathogenic. I mean, never in non-pathogenic strain. Well, this is the scheme. You remember, this is the scheme we follow to finally to identify this effector, and with the result we have with this analysis and with other analysis, and based on the specificity of the SOP-E3, we have designed a protocol to detect specifically Xanthomonas arboricola patobarpunae. Well, this is because, as I told you, this effector was found in non-pathogenic, always in pathogenic strain. And this was one of our genes that were defined as specific by genomics. This SOP3 is, I don't remember around here, but all of these are specific genes for virulence in Xanthomonas arboricola pathovarpurae. Well, we design primers as a probe and develop a real-time PCR protocol. We hybridize the primers and probes in silico with the genomes of Xanthomonas arboricola pathovarpurae and attach only were specific to Xanthomonas arboricola pathovarpurae, where this is the ABC primers and probe, and you see that it's hybridizing with many of the genomes available at this moment in Xanthomonas arboricola or even in other Xanthomonas. But as I told you, neither ABC primer nor SOB3 primers are really specific for Xanthomonas arboricola pathovarpurae. If you have a positive reaction with SOB3, you can have a Xanthomonas arboricola pathovarpurae, but you can have also a Xanthomonas species, but as I told you, it's always pathogenic. We never found any other non-pathogenic bacteria that was positive, that are positive with this primer. But if you are still using the ABC primer, you're going to have this combination. You have positive with the ABC primer, you're going to have Xanthomonas arboricola pathovarpurae, but you can have also these Xanthomonas that were isolated from prunus and are not pathogenic. I mean, at the end, the combination of both is going to give you a precise identification of the strain. It's going to give you if you have a problem or you don't have any problem. Well, this is just to show you that, just to show you that, this is the protocol we designed, and this protocol was provided to the Plant Protection Services and is used by the Plant Protection Services now for the screening they are doing in the nursery, greenhouse, and in the field. Going further, we did the same approach, doing some comparative analysis of the genomes with inside the Xanthomonas arboricola species, and we found or we have identified all the factors that are involved in the virulence of not just Xanthomonas from prunus, but also Xanthomonas arboricola from halsenut and also from walnut. And well, in the next future, we will develop protocols based on these results. And finally, because I have time for minutes, well, this is I talked to you about the past, I talked to you about the present past, and now I will tell you about the future. This is a really interesting model for plant pathologists in the next future. Well, actually, it's an interesting model right now. The HLV is well, most of you probably know about the HLV. HLV is Hualonbin is a disaster for citrus areas. You have this kind of symptoms, but the bacteria at the end kill the tree, and there are some areas of the world that cannot grow citrus anymore because they have HLV around. It's caused by Liberibacter, Candida Liberibacter. But Candida Liberibacter is also affecting other crops like carrots and also potato, producing the zebra chips. HLV and the zebra chip are quarantine disease also, and precise identification. In this case, it's even more difficult because this bacteria cannot be cultured in the lab. You have to work with uncultured bacteria. That this is for microbiology is a disaster. I mean, you cannot grow the bacteria on a plate. It's a disaster for us. But well, we need to work on that. This HLV and also Liberibacter, the diagnosis right now is based on most of them on ribosomal sequencing and on ribosomal sequences, and also if they are not ribosomal, that always general genes that are present in many bacteria. This means, at least in our opinion, that is not a good approach. It was a good approach when you didn't know about the genomes and you have no information about the genome. But now you have many genomes of this bacteria, and We know that there are also some non-pathogenic Liberibacter around, and they are not doing any symptom, and these non-Liberibacter bacteria are giving positive results with these approaches. The idea is in the future, or well, in the present future, because we are doing that now, is by genomics, by genomic comparison, try to develop the strategies for specific detection of this Liberibacter, but based always in factors that are related directly related with virulence, not using general genes that are present or may be present in many other bacteria. And well, this is a summary of what we are. We have the complete genomes, and the complete genomes give you information about the virulence factor that can be used for target detection tools. Give you clues about the about the infection mechanism, and all of them can be used for disease control. The massive sequence also allow you to direct detection of the pathogen and also to know the microbiome. And well, this is something that we can discuss later if you want. And the end is just to show that it's important in this kind of work, the people, of course, it's important the funding, but it's also important and very important facilities and resources. For this kind of analysis, we are using supercomputing centers, and we need bioinformatics, and we need help, because I told you that we are plant pathologists, we are not bioinformatics, and we are not molecular biologists. And especially it's also important to have an available database. I put here some of the ones that we are using, but we have to have this database at networks like the COSACTION and different resources that are essential for this kind of studies. Thank you very much for your attention. And that's
Thank you, Jamie, for your presentation. Next on the stage is Guillaume. Forget your last name. Sorry, Guillaume. Beloudou. Is that all right?
Yeah, that's right.
Yeah. From CFIA in Canada. We are slightly but surely venturing into larger and larger genomes. So next are also oomycete and fungal genomes. Yeah, a switch to introns as well.
Yeah.
So the stage is yours, Guillaume.
Thank you very much for the invitation for this meeting. So yeah, today I'm going to present on how we use genomics. So I talk on genome-enhanced detection and identification of plant pathogens. So I will start very quickly because I'm new with the group, so with a quick background of what I'm doing, my organization. After I will talk on genomic application in regulatory research at CFIA. So I will present first what we do using genomic for fungal identification and detection. So I'll present 2 cases. And I will present briefly the type of work we do in metagenomics and other related activities. So first, I'm working in Ottawa, the Canadian Food Inspection Agency, and our lab is called the Ottawa Plant Laboratory. So it's on Fallowfield, and we have different diagnostic and research groups. So we have a nematology section, genotyping and botany, molecular identification research group, seed science, entomology, plant pathology diagnostic, and my lab, the plant pathology research lab. So my lab, that is the Pathogen Identification Research Lab, so it's mainly to support the activities of the OPL for diagnostic section through research and technology of transfer. We provide scientists and other professionals with information support for regulatory decisions. And we work mainly on detection and identification of plant pests, fungi, and oomycetes of regulatory significance. And we work in forestry and agriculture. So the expertise of the lab is mostly on fungal detection and genotyping, mainly on Phytophthora, Verticillium, and other different molecular biology techniques as PCR, qPCR, isothermal amplification, macrosatellites, and more recently work more with genomics and metagenomics and some some work also with microfluidics. So we focus mainly our work on quarantine organisms and import-export material. So the work we're doing is based on Plant Protection Act and associated regulations. So the objectives are to prevent the introduction and spread within Canada of plant pests of quarantine significance, and to detect and control or eradicate designated plant pests in Canada, and finally to certify plant and plant products for domestic and export trade. So this morning we had a few presentations on genomics, and we show that in the last 2 decades we have remarkable advancements in genomics. Like Sébastien presented this morning, so the cost of sequencing is getting very, very low now. So we can sequence genome under $1,000, sometimes around $100 for some technology. And in the last few years, like at the federal government in Canada, we had access to a program called GRDI, so Genomic Research and Development Initiative. So it's allowed us to play a lot in genomics and to evaluate the limitations. So we have initiated here some projects where we've been able to evaluate how we can use those technologies. So I will start with how we use genomics for development of markers for fungal detection and identification. So with this project, we had 3 objectives. So the first one was to demonstrate a proof of concept for the high-throughput sequencing or next-generation sequencing, so that it was useful for detection and genotyping of target species. So also we can use it to develop specific markers that can be used for the plant pathology diagnostic lab. The second objective was to prove of concept to evaluate the pathways and the sampling methods as a source of target pathogenic fungi, to provide metagenomic info and identify hotspot area where that could be useful for the agency. And finally, is to support the diagnostic lab work, so with a new developed technique as qPCR or high-throughput sequencing method to facilitate the identification and detection. For some of you that may have worked mostly in mycology, so we use mainly as identification markers the ITS regions for the internal transcript spacer, and we use also the IGS for the intergenic spacer as the regions to differentiate different species of fungi. However, this marker doesn't work for all of the different fungal groups. So sometimes they have some species where it doesn't work, some subspecies, or we have complex species. So, okay, what is exactly this fungi? Is this species or this others? So in some of the cases, this specific region doesn't work very well and can't differentiate between the specific target organisms. So this is where having genomic resources can help us to do our work. So the genomic applied the next-generation sequencing methods and bioinformatics to sequence, assemble, and analyze function and structure of the genomes. So it provides tons of data that can be used to answer biological questions and to develop diagnostic assays. So now it's possible, the possible application in plant pathology could be on the development of large number of markers and assays, search for avirulence-resistant genes, do a large-scale identification and detection methods, and work with metagenomics. So this is the first case I want to present on the use of genomics for generating markers today. So it's a method that we name genome-enhanced detection and identification. So we name it the Jedi for the Star Wars fans. So basically, this example I'm presenting today have been published with Nicolas Faux and Richard Hamelin from UBC, University of British Columbia. So we developed a method where we can use genomic information and screen specific regions to develop specific markers. So the first step is to require access to assemble and annotated genomes of target organisms, but as well as for some other closely related species, taxa. So the genome of the target and related non-target taxa are required, like I just said. So we need to have some for the same genus or from the same order as possible. So moreover, sometimes they have some that are already available in the different database, like my colleagues present this morning. But sometimes we need to do de novo genome assembly, sequencing and assemblies. And also we need to have the full protein set that can be either produced and assembled for a target species under investigation and for a group of related species. Or we can get it from genome data repositories like NCBI. So they have, here they have more than 2,000 fungi genomes available on NCBI, but they have other databases available like the Microcosm and the 1,000 Fungal Genome Project, for example. So we have more and more access to different genomes for fungi now. So the next generation high-throughput sequencing technology constitutes a fast and cost-effective way to obtain whole genome sequences, particularly in eukaryote microorganisms. So this project has been developed on the TIGA project, a Genome Canada project. And just as example here on the graph, you can see the different groups, azomycete, ascomycete, and the different number of genome sequence available for different type of organisms. So some of them we have more than 100 genomes available, but others we have only 1 to few genomes. So the method that we use here was used to underline the principle of our method is to compare the protein content. So we need to do annotation of the genome and evaluate the protein contents. So within the genomes of phylogenetically related taxa, so the specific target and the related one, we insert the selection of targets that are discriminant toward the most closely related known species. So using bioinformatics pipeline, we divide our method in 4 modules. So the first one, that's here. So it's where we get the genome available from resources, where we get the genome sequence from resources, and we have annotation. So we prepare the genomic material and prepare the annotation. The module number 2, this is where we will discovering homologous gene cluster. So which genes are similar between genomes. And we look at them to find some putative unique genes for targets. So what regions will be unique to the specific target organisms. But at the same time, we also look about what is conserved. So what is conserved between the different groups. Like that, we can have a markers that will be identifying a specific cluster of groups together, and we will have some other markers that will detect specific target with unique regions. So we use this information, and after we use other bioinformatics pipeline when we do filtering. So we can check with the different database, oh, it is really unique, but or it could happen to other sequence organisms. So we do some cleaning with filtering with this module 3 here, and we do it for the set of unique species-specific gene, and also for the conserved group-specific genes. And the last step at the end, the module 4, it's about the designing of the assay. So this is some software that we use to design, example, some qPCR assays. So it works very well. So we use it in forest pathology. We were targeting some pathogen as Phytophthora, some Dotidiomycetes, and some Puccinales. So we try for some of them to design some specific markers that will be specifically targeting the genus, the species, and sometimes some lineage or specific clades. Just as an example here for Phytophthora ramorum, so we compare the genome of Phytophthora ramorum with Phytophthora infestans, Sojae, lateralis, Capsicis, Cinnamomi, hibernalis, and Foliorum. And when we compare them with the module 2, we've been able to get a number of 52,000 cluster gene regions. When we look about the one that were unique, we got a cluster about 1,600. And finally, we got at the end, with the filtration and cleaning, about 37 unique gene regions. From them, we have evaluated 28 of them for Phytophthora ramorum, and we have 5 of them that have worked very, very well and were very specific to Phytophthora ramorum. So it's a good way to develop quickly markers and to have also more than only 1 gene regions markers. Like here we have 5. When we have redundancy, we are more confident that the organism is present. So this was the example we developed with the JEDI, but we also, directly in the lab, we're developing a similar approach with a slightly different bioinformatics softwares to target other organisms. So we have identified some organisms where we need to use the genomic to develop tools. So we're targeting the Lachnelula wilcomi, the European large canker, Phytophthora ramorum, and Fusarium sporotrochiodes in P, and other organisms. So we're lucky in our lab at Fallowfield in Ottawa, we have access to different sequencing platforms. So we have access to the Ion Torrent, the PGM, and the S5. Those are the ones that I will talk more today. But we have also access to MySeq, Illumina, and Minion Oxford Nanopore. So very, very nice when we need to do de novo sequencing. The next step is to identify organisms that we need to target for work. So we do discussion with the Diagnostic Lab. So the Diagnostic Lab is my main client. So we work with them. What are the organisms they will need a specific assay? Which one they have complex species? Which one they need more info, or where the ITS doesn't work? So with the discussion with them, this is where we identify the species that we will work. So I put few examples here. So the test case that we used for this was the Lachnelula wilcomi, so causing the European large cankers. And this organism is causing cankers on large tree, and is present in Europe, Asia, but also in North America, as in Canada. So in Canada, it's only present in the Maritime, so on the east side. It's present in New Brunswick, Nova Scotia, and PEI, but it didn't move yet to the west. So this is why we're still regulating these organisms. So this organism is part of the Elochialis group, so from the genus Lachnelula. Most of the species are saprophytic, only few are parasitic, but Lachnelula wilcomi is one of the pathogenic. Mostly the identification of this one is by morphology and it's by eliminations, and it's very difficult to differentiate this one from the saprophyte Lachnelula occidentalis. Only few molecular assays have been developed in the past. Unpublished RPD was available, but it's very limited, and the ITS reagents works better mostly for the genus, but not for the species. So this is why we decided to work with these organisms, because it was tough. It's tough because first, it's difficult to extract the DNA. When you extract DNA of this organism, you have a lot of polysaccharide, so you don't get a good DNA quality and yield. Very difficult to do. And also, it's very difficult to differentiate from the closely related species. So it's okay. If it's worked with Lachnelula, probably it's going to work with all or other organisms after that. So we develop a different protocol to grow the pathogen on media. We rinse the media with sterile water to remove polysaccharide. And at the end, we've been able to get good quality, good DNA, and be able to start to do the library preparation. So these are the 7 Lachnelula species that we have sequences. So we sequence wilcomi, or target, and the other closely related one. And like you will see here, what is the most important, you see they have a number of contigs here. It's pretty high for wilcomi. It's 13,000. So it's not perfect. The best is to have the lowest number of contigs as possible, but here our goal was not to get the perfect genome assembly. It was to get enough genomic information to develop markers. So the goal here was obtained. So we got genomic informations. We have about 30 to 50 MB of genome sequence for all organisms. So we have a lot of materials to search for unique regions and develop specific markers. So the pipeline here that was developed by a group, I won't go in detail, but it's very, very similar to the one that has 4 modules before. So basically, it's the sequencing and the assembly first, like the module 1 before. It's an annotation pipeline that will identify the different protein and compare the homology of protein between all of them, the module 2. After it will be some quality search on other reference database, and finally the markers development. So very similar. After that, when it's the time to develop the markers, usually what we like a lot is to try to develop some primers and probe that will identify to the genus level and to the species level. So when it's possible to find some regions that will be common for all the genus, for example, but have different regions in the middle that will allow to do specific markers for each species. So when we're able to find those type of regions with the genome, this is what we do. Like that, if we have a negative sample from this target, we could know that, example, oh, yeah, this Phytophthora ramorum is not present, by example, but they have other Phytophthora species in that could cause a problem. So like that, it gives us more information about what may be causing the problem here. So we developed some tool using this approach. And we've been able to develop few qPCR assays very specific to the target. So this is 3 different regions. We have developed markers for like Nulla wicomii. So it was working very well. We got a sensitivity about 200 femtograms, so very sensitive. So the goal was was obtained. So we succeed on this. So was working with these organisms. So let's see if it can work with other organisms now. So we used a similar approach with Fusarium. So when we were talking with the diagnostic lab, they were interested about some assays for Fusarium sporotrochiodes, the one here, but also all the one in red, Lancetiae, Avesiarum, Graminearum, Oxysporum. So we look about which one has a genome available. And for sporotrochiodes, we sequence quickly the genome as well, assemble it, and use the pipeline. And we finish with very specific assays at the end. And we have also some assays that are developed for the genus level. So it was working very well. So I hope today I've been able to show you that the molecular marker development with Lacnellula species detection and identification was a great test case, and same for the other 3 pathogens, forest pathogen I presented before. We demonstrate the efficiency and efficacy of the whole genome sequencing for marker identification. It's a reliable approach for developing diagnostic tool for our agency, and it provides us with an established method for design molecular markers for other plant pathogens, DNA preparation, and bioinformatics pipeline. So now we have a better toolbox for this, and the focus on the development of molecular markers by exploiting the whole genome sequencing was rapid, sensitive, filled some gaps that we had, and the DNA yield and quality is no longer an issue. So it could address some bioinformatics bottleneck. The method is transferable to other pathogens. So I talk about Fusarium. We're working now with some Colloquium and some Tillacea species with this. And so we can be prepared for pathogen identification and diagnostic need. So the key word is preparedness. So this was the example of using genomics that we do. But because the theme of this meeting is about the era of metagenomics, I thought that it might be good if I present you some use that we do of metagenomics as well for our regulatory work. So we present before the genomics, but what is the metagenomics? So the metagenomics is the study of a genomic content in a complex mixtures of microorganisms. So the field of metagenomics is also being referred as environmental genomics, ecogenomics, and community genomics. But basically, it's the study of the genetic material recovered directly from an environmental samples. So the objective too, when we start to work in genomic here, it was could we use the proof of concept to identify the pathway and a sampling method for target organisms, and provide bioinformatics metagenomics info to help us to identify some hotspot area where we should do a better survey, more sampling. So surveying for fungi is very difficult. For insect, it's more easy. We can put a trap, you have attractant, the insect will be attracted, they will be catch. But for fungi, it's much more difficult. So we need to use some other methods. So the high-throughput sequencing is one of the ways to do it. So if we have environmental samples or a commodity, we can sequence with the sample with next-gen sequencing. We can do data analysis by informatics. After we look at reference collections and which one is matching, and we can make some regulatory decisions or recommendation on action. So the work I will present very, very briefly today is the work that I've been accomplished by Émilie Tremblay, that was a former student, PhD student in my lab. And she published 3 different papers related to the method and works that we have done on this area. So the first one is about the protocol that we have developed to screen for exotic forest pathogens using survey capacity using metagenomics. The second one is about evaluating the potential interaction with fungi, propagules, and insect caught in insect trap. And the last one is evaluating of the pollens pellet that could bring some potential spores of invasive species or invasive plant. So basically, now today, with the emerging forest disease caused by invasive alien species, represent an important threat to Canadian forestry and other country forestry, with the entry of spore material. So the international trade with the sawmill, compost, all the importation, all the import of live ornamental plant material, the environmental factors, wind, rain, airflow, insect vectors, phytopathogenic fungal, and all my seed spore spreading can be a threat for the different activity for the forestry. So they are difficult to identify due to their cryptic natures. So we need to better understand what is the source of introductions, what will be the origin of the country, what is the mode of introduction. Do we have any established molecular method that will help us to do biosurveillance? So we have only a few methods available, accurate and rapid to detect fungi, and but they are specific to specific targets only. The culture morphology and the host for identification can be very slow and sometimes non-specific. So we need to have a way to have some prevention of potential infestation. We need to be more proactive. So this is a project we initiated in forest pathology. So in discussion with the CFIA and the Canadian Forest Service, we decide of we try at the beginning to do a top 10. We finalize with the top 15, the top 17 of unwanted pathogens or forest pathogen that could be a major issue between our 2 organizations. So we have some exotic species like the one that we have in red there. We have some native one and some that were introduced in the country. And we decided, okay, let's work on those specific 17 species group first. Some of them are already causing more problem or are more targeted by the agency. We have, by example, the Phytosanitary causing the southern oak death, mostly in California and in UK. Causing severe oak mortality. But it can also attack other hosts, more than 100 plant hosts. It's a big nursery problems. Can attack to Sequoia, Douglas fir, rhododendron, camellia, vaccinium. We have quarantine measures between different countries on this. They can propagate via nurseries. So this is one of the organisms we target. And the second one is the oak wilt, caused by Brachyzerella fagesarum. This is a pathogen that is present in the US. It's very close to the border with Canada. It's just a few hundred meters to the border right now. So this is why we look at this one. So we include this one in this project to see if we can use metagenomics to find the presence of this organism. So basically the workflow, so I told you before that we have a good survey for insect, but not much for fungi. So what we have done here, we got access to different survey samples all over from Canada, from the west to the east. We got access to insect trap. So when they collect the insect in the insect trap, they use at the end of the trap, in the bucket, they use some propylene glycols to conserve the insect. They don't use it. So when they were surveying for the insect, they sent us all the juice of polyethylene glycol at the end, and we filter it on 0.45 microns, and we extract the DNA of that to look for fungi. We also add some spore trap that we set up more in Quebec and Ontario to get the spores that could be potentially from the air and from the rain. So we got those samples, this juice, the spore trap, we filter it, we extract the DNA. So after that, the next step was to do amplification. So we did amplification of specific regions. So the ITS1 regions for fungi and oomycetes, and the mitochondrial regions, ATP999 for Phytophthora. So we did the amplification of this, and we sequenced it with the ion torrent platform. And we did bioinformatics search after that to look about target and identification of the material. We also did the validation with some specific qPCR assays that we had available. So we did it for around 400 some different type of samples. Close to 300 were from spore trap, soil, and rotorod. And we have about 100 that were coming from insect trap. We sequenced it with PGM ion torrent method for 15 runs, and we get about 45 million sequence. Now with the S5 method, we can get this amount of sequence with only 2 runs. So we can get much more sequences with the new technology now. So these are the 3 regions that I just mentioned that we have amplified for the different target one. And we develop a bioinformatics pipeline to do a quality check and to identify the different organisms. Just briefly, the type of finding that we have found in the sample. Yes, we have found some Phytophthora species, but not the target regulated one. We also find presence of Heterobasidium species, some that we know that we have in the country, some that we didn't know that was in the specific regions. So now we need to do more survey and look more to look for symptoms and find them in the nature. So we have some genera, including phytopathogenic species that have been found in pollen samples. We have Fusarium, Ophiostoma, Peronospora, Phytophthora, and Pythium has been found. The other potential hosts for some, no potential hosts for some of the pathogen also have been found. We find some correlation from some insects that could vector some pathogens. Now we discover a new area where we need to refine sampling and inspections. And we have a better baseline now on potential presence of some organisms. The challenge we have to develop a pipeline, we have massive amount of data. Someone talked this morning about the tsunami of data that we have. The identification is sometimes impossible. Sometimes we can get only to the family or genus level. They could have some error that can don't allow to differentiate between some species. Database need to improve, and severity of result of invasive species are identified. Could have severe impact if we found one. So the technology here, use insect and spore trap, is fairly new. Can give us a good, potentially, survey method. Also the pollen and ODB for invasive species and plant could really help us to do it. We multiplex 3 different regions. The high throughput help us to do sequencing, help us to do it. We have now some guidance for CFIE biosurveillance survey. We have identified some potential hotspot area, high-risk area. We find some correlation with our next generation sequencing and qPCR. And we can do detection of closely related species that demonstrate the ability of resolve pathogen species. We are looking now to the transfer, to the potential of transfer the method to the diagnostic use for the future. And one thing I want to point very here is be careful, because this is a potential spore detection. So we found a sequence, and some people talk about that this morning. This is not a first report. When you have a first report, you need to have a pathogen. You need to be able to do a Koch postulates to declare it as a first detection, a first report. But we found a sequence, so potentially some spores. So it's the time to use it as a pre-screen and to go to look, do better survey, to send some people where you find the potential spore there, and to look about symptomatic tree, symptomatic plant, and try to do a better survey or better work. So just want to briefly, we still do a lot of work in genomic and metagenomics. So we have another GRDI multi-department project, Ecobiomics. We look about the soil and water microbiome. We have a Genome Canada project, BioSafe, where we use AmpliSeq to look at different genes that could be also involved with lineage, pathogenicity. We develop other project to develop some specific markers for target 1. We have a specific one where we have some survey from oak wilt, from oak log that we import from US. We have put some specific insect trap here. We try to look if we can use this tool to detect some potentially the oak wilt. And I briefly mentioned the use of the pollens. So now we have new project with pollens and bee. So we will look about the potential of finding some viruses, bacteria, fungi, and plant. So we are evaluating the use of this tool, but not just for plant pathogen, but also for the bee health, also some pathogen that could infect the bees. So I want to thank all the teams that helped me for this one. We talked about bioinformaticians before. They are really crucial. They're very important. They play a big role in this one. With this massive amount of data, we need them. So I think we need to continue to push to keep them and recruit more bioinformaticians. And I just want to thank all the funding I got for this project. Thank you.
Thank you, Guillaume, for that very nice overview of different types of applications. So with that, we actually are now at our first discussion session. So this would be an opportunity to, first of all, raise questions, specific questions to the authors of the talks we've heard so far. And otherwise, we've been hearing about applications in virology, in bacteriology, fungi and oomycetes. So one of the things is there are many applications, possible applications for next-gen sequencing or high-throughput sequencing. Is it too much? All these different applications will cause new types of work. Can we manage? Can we make sure to keep the entire community on board? So what are the challenges for us to address? And I would like to open the floor to all of you to comment on that. Yeah. Baldi, you would like to kick off? Oh, Sebastian.
Just one very important point is this plant health bioinformatic network in that case, but to have this new community of people. And one of the axes is also to make training material. So in that case, the goal is we start with viruses because initially there was a majority of virologists there, but we try to have a combination of having training material, having mock data to play with, so people can train progressively and understand. It's complicated, but it's doable because we have seen through cost action that in 4 years you get plenty of people really leveraging significantly their understanding of bioinformatics. We do not need to code this, just using smartly the existing tools that are more and more user-friendly.
So a good comment, yeah, definitely. Baldissera just made a good comment as well. So to further facilitate this discussion, it would be good to have the speakers of this morning's session on stage. So if you could please join me here on stage now, that would be maybe good. Yeah. Yeah, so training sets, opening up collections with associated metadata, key to generate reliable data sets and to mobilize the field. So are there any questions from the audience to further trigger this discussion? Challenges for your own lab or maybe, gaps?
We talk about training sets. I just want to say where we need to work a little bit more here based on our works is what is the QC, QA that we can do from this. So we need to train people, make sure that we have some different steps when we do bioinformatics, when we use this approach to make sure that, yes, at the end we got this name there. What is the step? But one of the challenges in this is the different program and software are evolving very, very quickly. one method that is used now. They already have other methods that use and better software to use this. So when you standardize something, most of the time they have other techniques now that seems to be more quicker, do more filtering. That is this, and this will be the challenge with this one because, example, a qPCR, we stay many years with the similar approach, but on bioinformatics here, the knowledge is always continuing, and now we have the artificial intelligence learning machine that could eventually help us on this. So yes, we will need to make some very specific QA QC method, but because the evolution of this continue and is very quick, so this is one of the challenges that we have.
Yes, so indeed, and so there are also some of these initiatives are being addressed under one of the Euphresco projects, as I am correctly. So on the virus collectors, also some of the QA QC elements included, but does that also extend controls in the routine sense as we see them for qPCR or other PCR tests?
Yeah, there is an Euphresco project starting, but there is also an ongoing H2020 project called Valley Test, and one of the tasks there is to develop guidelines for use of high-throughput sequencing in diagnostic, so including this issue of quality control, quality assurance, which kind of control exists, which step of the protocol, both in the lab and in bioinformatics, needs to be completed, and indeed a new issue is the bridging that we call bridging between version 4 and version 5 of the kits, or between version 6.2 and 6.3 of the software, indeed.
Yeah.
On a different topic, I see now that this community is developing diagnostics for these huge numbers of species. But what I don't see in the databases is where you've got multi-species database covering a lot of different genomes, that actually the indicative genes now, rather than the ITS, are being marked up to say that this is the gene that is being used for a species diagnostics, and that it can be easily searched for, and where indeed other sequences in the genome are being used more on the variome, and therefore you can identify different strains within a species. So is there an initiative somewhere, locally, regionally or globally, to actually try and assimilate all this data on the diagnostic markers for each species, so that they're eminently findable, and others can also then further use this information for other species?
So personally, I'm not aware of any of those initiatives currently. So in the past, we had a EU project focusing on barcode regions. So that was the QBall project. So under that project, there were barcode regions identified for quarantine species at the level of regulation. So that's quite often under a species level, so part of our subspecies, whatever. But I do agree that at some point, broadening the scope of that would be very, very useful and needed, actually. So a sort of plant health genome initiative, sequence all regulated pests and lookalikes, closely related species. And there's already quite a large number of genomes sequenced out there, but I think the larger the genomes, the fewer the reference genomes that are available, and also the quality of those genomes come into play at some point, of course. Because if you're looking, for instance, at species-specific gene content, but you have only one strain of a genome of one genome sequenced, how complete is that genome? Is a certain gene actually missing from the genome, or is it just missed in the assembly? And so a redundancy in strains is very essential in that sense to build reliable data sets.
And can I also just follow up? I mean, to do that and enter it into a genome sequencing or into the genomes, I mean, the type of reporting page that you may require as labs will be quite different from somebody just depositing a gene when they've done a functional analysis as to, as you were saying, the who, what, why, when, you know, and where. And so that would require a dialogue with the major genome providers as to what you would need to ensure that the entries that went in were as flexible and usable as possible for multiple purposes.
Yeah, the answering your question, for instance, in bacteria, is specifically from each species. I mean, for instance, in case of Xanthomonas, we have an ACOS action that now is collecting information about these effectors I told you about the virulence factor, and we are doing a collection of a database of this effector that would be available for the people. But I don't think that is for bacteria. This is what I know. I don't think that is a general thing. I mean, you can try to do something general for bacteria, but you have to differentiate between species to get this information of a particular effector or particular factor that involve virulence.
Thank you, Bert. All presentations are very interesting. The molecular methods rapidly develop. Whole my life, I organized the work of plant quarantine diagnostic laboratories. And from one side, molecular methods is a great step forward. From the other side, for me, these methods bring us many problems in future. I want to ask all speakers, what do you think about the future of routine quarantine diagnostic laboratories?
I think the need of still using the regular method with morphology, with a test, it will still be a need. I think where these tools, while I see the use directly in the diagnostic lab, it's mostly when you need to do more high-throughput sample processing. You have so many samples to test, or if sometime you get to some species or subspecies level very specific, and sometimes the current method that you have don't allow you to do it, or you need to do 10,000 plates to being able to get to this level. So this is why some of those tools is helping to do this. Like I just mentioned before, we got the sequence. We need to be careful because we don't have an organism in hands with when it's just a sequence. So yes, it can bring some challenge, but I think at some point this can be used as a pre-screen at some time to help the identification and the process. And after you have all the other methods that could help you to make sure that it's really the target one.
From my perspective as somebody that runs a diagnostic virology lab under an NPPO, I think the question is how we integrate high-throughput sequencing within our toolbox of diagnostic tools to get the best effect for the best cost. And some of that will be dealing with samples where we have diseases of unknown etiology as we're using it now. Some of it will be how we move to use high-throughput sequencing as a surveillance tool. So how can we do bigger samples, higher throughput, and do big field samples through the sequencer in the similar way to the way we're doing the viromic samples and looking at environmental, ecological type questions, but can we do that from a plant health sense? As we do that, what we'll find is, as we're finding even with single sample diagnosis, we flush out more and more unknowns, all things that we didn't know were present. And that then brings the question of how we share that data and how we collaborate to take away some of the fear of finding those things. So if we take the Actinidia plantago virus that I showed, that originally started as a conversation between ourselves and John Hammond at the USDA, because the plantago virus was actually the virus he worked on for his PhD in the 1970s. That then spread to Marlene at NVWA in the Netherlands, who went looking at plantago. We went looking at plantago. We got the original isolates out. And so we knew we had this virus that we then sequenced and showed it was linked to the virus in New Zealand that Arno found in 2013. Talking to Sebastian this morning, that story may carry on building. But what you then have is a question of how we get the right people together to share the data we have to show that this material is potentially everywhere already, it's of minimal threat, or it may be a major threat. And it's how we get those casual networks built and linked so that we have a more routine way of sharing that data, so that before we actually publish, we can say, well, actually, we think this is widespread, or actually, we think this is something to worry about.
Thanks. Piero Bianco from Milan. As a very interesting presentation, thank you to all of you. And but my question is opens maybe another issue. Jaime Cubero spoke about, said that the microbiome may modulate the disease. So, and is an incoming, is a new issue, but this is a concept of pathobiome that in other, so that, is, that from the scientific point of view is interesting, may imply a very uncomfortable field because it means that it's not enough to detect the pathogen, but also to consider all the microbiome. So it means that these techniques, the genome-based detection may help in this, but I think that's we need more and more powerful tools, and also, so it's a very tricky.
Yeah, issue. Well, I only partially agree about that. I mean, when you have a pathogen and and it's clear that the pathogen is producing symptoms, like one of the bacteria I showed you, the citrus canker, if you have citrus canker, you're going to have the disease. But if we don't know yet, and we didn't study it yet, but the plant health maybe depend on the microbiome, and this much sensitive to the pathogen is with a certain part microbiome than the other. This is one thing. I mean, it's clear if you have a positive with positive citrus canker is because the plant is infected by the bacteria. Another thing is that some of these diseases that we don't know yet if are produced by one specific pathogen, and this is a mix of the microbiome. This is true, but yeah.
Or some pathogens that sometimes are silent, are not. Are not. So the disease is not evident, but the pathogen.
Yeah, that's I completely agree. Yeah.
So phytoplasma, some bacterial viruses.
Yeah.
Thank you.
I believe, Françoise, you wanted to comment on the previous topic that we were talking about, data sharing and making available these genomes.
Yes, I think it's a comment. I have two comments. Most of regulatory diagnostic is now done under accreditation. So with the system of quick evolution, that's something that needs to be taken into account. So if the system changes all the time, how can you maintain accreditation? Is HTS possible for accreditation, even given how quickly it develops? So that's a new system, I think it's not only valid in plant health, it's valid in medical analysis. So we have maybe to learn how they are using it. So that would be a first comment. My second comment on the storage and the validity. Bart, you referred to QBall. So QBall in particular was referencing all the sequences produced during the project in a specific database. And the specific database is now maintained in EPPO, but that's said in passing. But there was a need. What I want to say is that there was a need to have a database where you're certain about the identification. So you are sure you're linking sequences to a specimen and to a well-described and identified specimen. So for regulatory purposes, again, I'm insisting on that. My intervention is in that context. We may also see the need to have curated information, and not only information that you find in any type of database. And I think we really need to think about it. QBank at the moment is not, this is part of the evolution, but we've not, for the moment, established anything. But the curation, one thing, and is do you see some, do you have any ideas about that?
Can I just pick up the validation point? So I think, as ever, the validation required will depend on the scope of your test. So if you are using it for screening for knowns, so thinking about the import requirement, the export requirement, then I think you can approach validation in the traditional sense. You know what you need to find, you can do your limits of detection, you can do your range of detection. I think where it gets difficult is where you're doing it on single sample diagnosis, dealing with diseases of unknown etiology, and you are likely to find something that isn't in a database, is badly characterized. And I think then we have to go back to thinking about validation at the systems level. So paper by Rowenhorst in EPPO Bulletin, where we talk about layering tests. So you're validating your specific finding rather than technology itself.
In food microbiology, they are to test level now because usually, example with bacteria, they usually only deal with maybe 5 to 10 bacteria only, so they use the full genome of this one. But in plant pathology, is more challenges because we deal with so many different organisms. So this is the big challenges. So for now, we still use a curated method with 1 markers, but how the technology is advancing, I think, and maybe in the future, we will get more full genome of eukaryotes at one point. This is the challenge because they're big genome. I think maybe in virology, it will come maybe quicker than on the eukaryote side, I would say. But we're moving to that, to see the QA, make sure we have curated database of all of them. But it is the big challenge in plant pathology because the number of organisms that we deal is very large. So.
And just to quickly follow up on that, so I think especially for eukaryotes, an organized, coordinated initiative to start sequencing genomes, and not only sequencing them, but annotating them structurally and functionally, because that is what we'll need if we want to start building robust detection pipelines or other predictive tools. So, and yeah, so the risk of duplicating work and the cost that is associated with that is maybe even larger for eukaryotic genomes than for maybe bacteriological or virological genomes. So it would be good to think about that. So we have room for one last question. Chris, yeah. Oh, 2 then, okay.
Okay, I was thinking of, there's quite some NPPOs present here as well, and the fact that Adrian also mentioned that we more and more have a lot of findings of new viruses, unknown viruses, not described viruses, or any other pathogens. It must be quite worrying with the new legislation coming up, what to do with that, because it's we'll have a new list of quarantine pathogens, RNQP pathogens, but if no or little biological information is available and they're sitting at our desks in the lab, it must be quite worrying for them as well, what this actually means to the different countries. So I think we need some good interaction with the NPPOs as well, and especially those labs that have or are a bit more distant from the NPPOs, what to do with that information.
So from our perspective, since we started using high-throughput sequencing in a relatively routine way, and we're running a MySeq run of 24 samples around every 6 weeks now. And that tends to be the difficult to describe things. It tends to be the ones where we've got symptoms on test plants and can't diagnose it any other way. And we have a semi-routine meeting with our risk managers where we go through everything that's been on the sequencer, what's come out of it, including the fragments and the question marks. And we simply go through that on a list-by-list basis and say, to be honest, this material is import, it's something funny, it's probably not worth worrying about, it's already been stopped. You then get this is from domestic material, but it's from a group of viruses that are known to be cryptic. Again, it's probably not worth worrying about. And then over that period of time of several years, we then got to the point that when we found the EUCAS, for instance, we then said, actually, this is probably something to worry about. And at that point, you've built that trust relationship between the lab and the NPPO, that when you say, actually, this is probably something to be concerned with, that you get the action, rather than, as you say, every single week, if there's something new, you almost get immune to it.
Yeah, of course, if there's a good interaction with the NPPO, this is what happens. But I can imagine that some labs do not have that interaction, and then it becomes a bit more worrying, I think.
But I think there is a parallel in interest between the researchers and the NPPOs, in the sense that the researchers also would like to work on the most interesting unknown viruses they see. And I think Sebastian described this process also from the research side. And I think what just was described, the sitting together or making a selection, there is a need for the researchers also to make a selection. If you find 200 new viruses, which one are you going to work on? And that could be done in collaboration with the NPPOs to see what's the most interesting, as Adrian just described.
Don't forget also that you're working on DNA, not on organism. When you sequence like dirty sample from the environment, you're sequencing DNA only. It doesn't mean that the organism is alive and causing diseases. So you basically have data, raw data that needs to be validated afterwards. And it's following up your question about using genomics or metagenomics everywhere. Should be considered as a tool, like a guide, you know, to follow up validation steps, to say for diagnostics, this is this organism. It's not the ultimate answer to what's in the sample, but if you're going to still using the PCR and other techniques that has been set up 10 years, 15 years ago to validate all of these, and you have to stick with the biological meaning of your findings, not only on the raw data of your findings.
Just to pick up on that, so we're saying that we're using tests developed 15 years ago, but we've heard about the use of Cox postulates this morning, and that is essentially a test that was set up 150 years ago. So do we also need to review how we address causation and different ways? So when you've got something that isn't a single pathogen, single disease issue, and you have a multi-microbial disease, that you can actually address it in a more meaningful way.
Okay, I would like to end the discussion here for now, so we can break for lunch. We'll meet together here at 1:30, and then we'll have the final two presentations, and then still around for discussions, so make sure to think of some cool discussions items to be discussed. Yeah.